Hello everyone, my name is Oluwalademi Adepoju, and today I will be describing my research project surrounding the expression, purification, and characterization of the recombinant heparin sulfate glucuronyl epimerase, also known as the C5 epimerase enzyme. I carried out my experiments with the help and guidance of my mentors, Dr. Kubi Balaguranathan and Dr. Yukio Saijo. I will also present the results we have gathered as well as the next steps we will be taking for this project. Firstly, I think it's important to talk a little bit about heparin. Heparin is a blood anticoagulant commonly used in medical procedures such as surgeries and dialysis. It is traditionally extracted from animal tissues, but that causes inconsistencies in production and raises concerns surrounding contamination risks. A more reliable and perhaps safer approach is laboratory-based heparin synthesis, which reduces risks of contamination and is comparably less variable in its availability. This procedure, however, requires C5 epimerase as well as other enzymes to complete its biosynthetic pathway. Now let's talk about why that is. Heparin is comprised of repeating units of either iduronic or glucuronic acid and glucosamine. The naturally occurring form of glucuronic acid needs to be converted to iduronic acid for increased flexibility of the heparin chain and enhanced interactions with proteins involved in the anticoagulant activity. This conversion is catalyzed by the C5 epimerase enzyme, which shows that the enzyme is essential for the biosynthetic pathway of heparin. Beginning with the procedures that we carried out in the lab, the C5 epimerase gene was first extracted from a plasmid in my mentor's lab and then sent for sequencing to ensure it was the right gene. The plasmid we were going to use was also sent out for sequencing, and so we were able to choose two restriction enzymes, XHO1 and HIND3. As you can see in the simulation of the recombinant plasmid above, XHO1 and HIND3 produce a fragment that corresponds to the size of the C5 epimerase. We also ordered the primers for the next step using the restriction site sequences. We began cloning the gene through the above processes. We first amplified the gene through PCR and purified it using gel electrophoresis, then digested both the inserts and the plasmid with the restriction enzymes and ligated them. Subsequently, we transformed E. coli cells with the recombinant plasmid, plated them in an LB-AMP plate, and cultured 12 colonies, after which we purified plasmids. We first cultured in a small volume to confirm the presence of the insert using the second digestion with PST1. And as you can see in the diagram, the enzyme PST1 has restriction sites in both the inserts and in the plasmid, so it was a suitable enzyme to use for this process. We carried out gel electrophoresis and used our knowledge about the plasmid and insert sizes to confirm their presence. The above picture is the image taken afterwards, with the band around 6,000 base pairs being in the plasmid and the band around 2,000 base pairs representing the insert. Due to some issues with the gel, we weren't able to analyze number 1 to 3, but from this we chose number 4 and number 5 for large-scale culture. We expanded the cultures for number 4 and number 5, after which we carried out endotoxin-free plasmid purification in order to start transfection. We measured the concentration of the purified plasmid afterwards. We found the low concentration of number 4 puzzling and decided to carry out gel electrophoresis once again. We first digested number 5 using PST1 again and ran number 4, undigested number 5, and digested number 5 in the gel, producing the image you see on the screen. The higher band in number 4 and both number 5 samples represent the plasmid. The middle band in digested number 5 represents the expected fragment produced by the PST1 enzyme. However, we were puzzled by the appearance of the lowest band as we were unsure of what it could be. So we sent undigested number 5 for sequencing. Sequencing results provided an answer to this, as we observed extra sequences at the 3' end of one of the clones, but the other clone showed the expected sequence. We have begun transfection and culturing of cells using the ex vivo mammalian cell system. The next steps for this project include the purification and characterization of the recombinant enzyme, and finally, we will evaluate the activity of the enzyme and demonstrate its function using NMR and mass spectrometry. We've come to the end of my presentation, and I would like to thank you all for listening to my presentation. Please do not refrain from asking any questions. Thank you and have a good day.